Good morning. So in my presentation, I'm going to talk about regulatory regions which link retinoid and vitamin D signaling. So in my presentation, I will talk about the introduction of for retinoids and functional genomics in two parts, and in the last part, I will describe our results. So I think most of you heard about Albert Szent-György's definition. So a vitamin is a substance that makes you ill if you don't eat it. So indeed, a series of researchers identified the link between the diseases and the vitamins. Moreover, in the last more than 120 years, the researchers identified the structure, the metabolism, the physiological roles of vitamins, including the lipid-soluble vitamins. So in our study, we worked mainly with retinoids and vitamin D. So vitamin A actually is not a single molecule, but a group of related lipid-soluble compounds. And for this reason, actually, sometimes we prefer using the term of retinoids instead of vitamin A, because some of these molecules are synthesized in laboratories or factories, used in skin therapy, for example, and also in research. So vitamin A is essential for vision, reproduction, immunity, embryonic development, and so on and so on. And vitamin D is also refers to multiple molecules which are related. So basically the biological active form is the 1-alpha-25-dehydroxyvitamin D3, so this is the calcitriol, but many other molecules are also considered or named as vitamin D because they have similar function, or they may have been converted into the active form. Adequate vitamin D levels are essential for calcium and phosphate homeostasis, supporting bone mineralization, and also it turned out that also vitamin D is essential for immune functions. So in many cases, vitamin D can increase the level of proteins which are essential for antimicrobial defense. So to understand how these molecules work, I just would like to show you some important regions and molecules which are responsible for the transcriptional regulation. So in our genome, the genes are under the control of so-called gene regulatory regions, such as the promoters and also distal gene regulatory regions. And these regions are occupied by different proteins, protein complexes, including the RNA polymerase II in this case, which is responsible for the transcription. And also promoter region and also distal regulatory regions are occupied by different transcription factors. Transcription factors are proteins which can recognize different DNA sequences. And I haven't mentioned, but retinoic acid receptor and vitamin D receptor are in fact transcription factors. So they have dual function. They can detect the presence of the ligand, and at the same time, they can recognize certain genomic regions which contain their specific DNA sequences, and in this way, they can regulate the transcription. So basically, these proteins are important in mediating the effect of the ligands of retinoic acid or vitamin D. What happens upon ligand binding? These transcription factors, retinoic acid receptor and vitamin D receptor, start to recruit other proteins. They are called transcriptional cofactors, including the mediator complex, but also many other factors are recruited by these transcription factors, and they initiate the transcription by modifying the activity of RNA polymerase and so on and so on. I wouldn't go into details. So what we should understand here that, I'm just going back, sorry. So upon ligand treatment, the transcription factors recruit cofactors. they activate the RNA polymerase and the chromatin environment, and the transcription is initiated. Therefore, messenger RNA is synthesized, transcribed, and these messenger RNA molecules are used for protein synthesis, and they contribute to the cellular response of the ligand. So our question was whether there is interaction between the two types of receptor, retinoic acid receptor and vitamin D receptor, how they can act together, how they can cooperate at the level of the chromatin. So my second part will be about the functional genomics. So functional genomics actually is study of genes and intergenic regulatory regions of the genome, which contribute to different biological processes. We want to understand how genes can be turned on and turned off, which proteins are responsible for this regulation, how the cell can respond to external or internal stimuli. We actually use multiple strategies, multiple methods. They typically global approaches, so we investigate events genome-wide, multiple genomic regions, multiple proteins are investigated at the same time. So as a consequence, typically we are facing challenges in processing, interpreting, and integrating data sets. So instead of the problem of how to collect data, the main problem here or main challenge is how to understand, how to interpret the data we generated. So here you can see a genome browser view of a typical functional genomics data set. So what you can see here on the top, you can see the gene. So on the, let's say on the x-axis, you can see the different genomic regions. This is the gene which is under the control of multiple transcription factor. So on this figure, you can see different tracks, and these tracks actually show so many things. I wouldn't go into details. I just would like to highlight that actually you can compare the genomic regions, how they contribute to the gene expression, the regulation. Also, you can see the openness of the chromatin. So many things can be investigated in the same time using these global approaches. So focusing on questions. So using these global approaches, RNA sequencing and chip sequencing, we could identify that almost 100 genes are regulated by both type of receptors. So we use a special synthetic agonist, retinoic acid receptor ligand, and the vitamin D. So in this way, we could identify the genes which are regulated together by these two ligands. Similarly, we could identify the binding regions which are occupied by two types of receptors. Moreover, we could identify the genes which are regulated together. So the blue indicates the retinoic acid receptor ligand, the red one is the vitamin D, and with gray you can see the combined treatment when we added the two ligands together. So in many cases, there was cooperation between the two ligands. So we could identify these common binding regions of the two receptors, and actually we could show, in many cases, we could identify the motifs, the short DNA sequences which are responsible for the co-binding of the two receptors. Previously I mentioned that it was not clear how the two receptors can cooperate, how they can work together. And actually, our candidate was the mediated complex, which can be considered as an indicator of the activity of the regulatory region, because it's a very important activator of the polymerase complex. So we investigated the level of the mediated complex in these regions. So here you can see the co-binding of the two receptors, retinoic acid receptor and vitamin D receptor. The peaks actually indicate the presence of the proteins, and here you can see the level of mediated complex, MED1. So vehicle, let's say this is the control, AM-treated cells, retinoic acid receptor ligand, vitamin D, and the combined treatment, and you can see, so briefly, the highest peak can be identified in the case of the combined treatment, indicating that in these cases, at this specific region, there is cooperation between the two ligands. So in summary, we could, in our research, we could identify common binding regions of the two receptors, retinoic acid receptor and vitamin D receptor, and we observed that at these regions there might be cooperation between the two receptors, so they work together to recruit cofactors, transcriptional cofactors, and as a consequence, using RNA sequencing, we could show that actually, in the case of the combined treatment, the transcript level is higher, meaning that in these regions, in these genes are under the control of both ligands, and they act in a cooperative manner. So I would like to acknowledge many of our colleagues, especially Andrés Amianeshez and Laurent Götzi. They were responsible for performing the experiments and do the transcriptional and bioinformatics analysis. And thank you for your attention.
Thank you very much. Now we have also time for maybe one or two questions from the audience. Your talk was very impressive, actually, and inspired a number of questions, I think, in most of us. But I would like to simplify the case. You introduced the vitamins in general, that if you don't have one or another, then we would have a disease. Now, what you are showing is that the story is a little more complicated because sometimes they interact. And so maybe we need at a certain place in the cell, not one, but at least two, to have maximal effect. Does it say that if we have vitamin deficiency in combination, we would have a different type of disease, or it means that if we have a deficiency for one vitamin, it can be substituted by another one?
That's a very good question. So as you could see, basically they have distinct but overlapping transcriptional programs, meaning that in some aspects they work together, but they have their specific functions. So for the gene regulation, you can see this kind of interaction, but you do not see necessarily the consequences of the combined treatment at the cellular level. So it's hard to dissect. I mean, it has been shown for many transcription factors that they work together. Interferon beta is under the control of multiple transcription factors, and so on and so on. But it hasn't been shown that these two receptors can work together. So basically, it was rather a mechanistic question than what is the consequence of one or the other, the lack of one or the other.
Please forgive me, I'm a physiologist, and so I would like to interpret always this in a broader context. And if I may add an additional question, do you believe that this kind of interaction has an evolutionary aspect?
It's a good question. I mean, it has been shown that there is evolution, so similarly to the proteins, there is evolution of regulatory regions. So basically, if you see something happening in the genome, it happened with the reason. So obviously, mutations can generate this kind of recognition sites or DNA motifs. And obviously, it turned out, so if you investigate the binding regions of different receptors, in many cases, you can see this kind of hotspots of regulatory regions. So somehow the transcription factors prefer to bind close to each other. So if you investigate many transcription factors, it seems that there is some trend of forming these motif-rich genomic regions which are responsible. So it didn't happen by chance, for sure, but what is the mechanism or the route which lead to this kind of regulatory hotspots? I don't know.
Thank you very much. Any other question from the audience, maybe? In the meantime, if not, then we invite you to come forward to the center of the podium so that we can have a group photo.